Today's video, we will explain the structure of primary to quaternary proteins, as well as the forces that hold them together. So at the end of the day, a protein's structure and function is determined by the primary structure. Primary structure is the sequence of amino acids. Amino acids are connected by peptide bonds, which are also the amides. The number of amino acids will determine the name of your peptide. If you have 3 amino acids, you have a tripeptide connected by 2 peptide bonds. Now the amino acids are also called the R groups or the side chains. Sometimes it might be referred to as a residue, but the only difference with that is the residue will also include the N-terminus if you're talking about the amino acid on the end of the peptide chain. Okay, so keep in mind that the sequence of amino acids are written from the N to the C-terminus. And the primary structure is planar, rigid. Rigid means that there is no rotation, no bond rotation around where the resonance occurs. So be really aware of this vocabulary because the exam, they might test you how many charged side chains are there versus how many charged functional groups are there. All right, so the side chain does not include the terminus, but if they ask charged functional groups, then that would include the terminus. In secondary structures, we are concerned about the spatial arrangement of the polypeptide backbone. So a secondary structure consists of regular and irregular structures, which are the loops. The regular structures are the A helix and the B sheets. The B sheets can be parallel, so parallel C and N terminus, or anti-parallel. The forces that hold the secondary structure together is the hydrogen bonding between the carbonyls and the NH. In A helixes, this hydrogen bonding occurs within the helix, whereas in B sheets, the hydrogen bonding occurs between different strands. And also, the R groups in the alpha helix are all sticking outside the helix, so it's outside. Whereas, in the B sheets, the R groups will alternate above and below the sheets. And also in alpha helix, the carbonyl oxygen will hydrogen bond with NH for residues downstream. Okay. Now we'll talk about tertiary structures, which is the arrangement of secondary structures or side chains. On the exam, they may ask you to look at a picture and tell you what structure it is. If you see one helix or one sheet, then it's a secondary structure. But if you see a jumble of mess, then it's a tertiary structure because it's an arrangement of secondary structures. But if you do see a compact structure that seems to have a core, then you can know it's a globular tertiary structure. So this is compact and soluble, whereas if you see something more linear, it's a fibrous tertiary structure that is insoluble. So there are 2 types. There are the domains, which is when you look at a picture and you see a cluster and a core, so there it's compact. That would be a domain. And if you do see several secondary structures together, but they are not making a hydrophobic core, and they don't seem to have that distinct structural unit, then you can know that it's a motif. Another note about tertiary structures is that In the core, you have everything that is hydrophobic, whereas outside, you have everything that is hydrophilic. So everything that's outside may be your soluble or hydrophilic side chains, such as the amino acids, or your secondary irregular structures. Everything inside will be the hydrophobic side chains, such as nonpolar amino acids, as well as the regular secondary structures, such as alpha helix and beta sheet. Okay, so outside is philic, inside is hydrophobic. Now let's talk about the forces that hold tertiary and quaternary structures together. The main driving force that holds the structure is the hydrophobic force, whereas recall in secondary structure, it's the hydrogen bond. So this is a main force. There are other minor stabilizing forces, such as hydrogen bond between the side chains or the backbone, salt bridge, also called ion pairs, which is a non-covalent electrostatic forces between positive and negative groups. So this is pH dependent. So positive groups include lysine and arginine. Negative groups include aspartate, glutamate, and depending on your pH, you could also consider histidine being in its protonated form, or tyrosine, its hydroxyl being deprotonated, or cysteine. And another stabilizing force is a disulfide bond. So this is a covalent bond between cysteines. Okay, now let's talk about, so these are stabilizing forces, and this is a main force. Let's talk about manipulating the factors to denature or unfold these globular proteins. If you add heat or some detergent, you can disrupt the hydrophobic force. Heat can also disrupt the hydrogen bond. Or you could change up the pH, which will disrupt the hydrogen bond and the salt bridge. Salt bridge or ion pairs, different names. And this is because if you change the pH, some of these groups will change its protonation state, so that will affect the ion pairs. pH, all right? Or you can also manipulate the salt, which will manipulate your salt bridge force. And also you can use reducing agent to disrupt disulfide bonds. All right. Now, last thing I want to note is a quaternary structure, which has the same stabilizing forces, as a tertiary structure. Quaternary structures may be named as dimers, trimers, etc. And the prefix just indicates how many subunits there are. A subunit is also called each polypeptide chain. And if you have identical subunits, it's called a homodimer. And non-identical subunits are called a heterodimer.